All right. Actually, we have, since there's three of us talking, and I want to save a couple minutes for any questions that might come up, I fear we'll get started. So I'm gonna start kick us off today. Let me just see if I'm taking controls and everything's going beautiful. I want to start actually this current release that we are going to be talking about today, and the portal that Maria and Doug are going to speak about, really is the effort of this team at the Broad. And I wanted to particularly call it out because this is a uniquely big release that we all did to erect a brand new software platform, make it publicly accessible, convert our old portals over to this new UI, and really just a treasure trove of data is in the new portal. And also I wanted to just send a special thank you and a shout out to two team members who have left us in recent months. Many of you may know them from various email communications. Marcin's been part of our team since the beginning, before we had a portal. And he just left to go to Takata. We'll miss him. And he actually has been responsible for so much of the data that we've ingested over the years. Ben Alexander really was instrumental in building like the first actual portal that we had. So these people were instrumental in building the huge AMP resource, but we'll miss them and we're very happy for them. Ben's at Pfizer and we still hear from them occasionally. So we'll miss them, but I wanted to thank them up front and say that we're really happy for them, but we'll miss them. All right, so today, we have three topics, actually two major topics. I want to kind of quickly give you a reorientation. Last time, for those of you who are on our webinar last time, we talked about the Common Metabolic Disease Knowledge Portal. Since then, we've officially launched it. We had some nice press from the Broad and NIDDK and the FNAH about it. We launched it in tandem with ASHG because we couldn't be there in person this year and do our usual booth and exhibiting. But what we were able to do is make this publicly accessible, and it now replaces all of the common metabolic disease knowledge portals. So I'll talk about that in a second. So I'll give you a little bit of a preview, a reminder of what's in there and how you can use it to start off, especially because it will help sort of volley into what Doug and Marie are going to talk about, which is the Musculoskeletal Knowledge Portal, or MSK-KP. And Doug's going to give us a vision for this. He really was instrumental in making this happen for us and really sort of represents the musculoskeletal community in terms of their needs and wants. And I think it'd be really good to hear from him as to why this portal was necessary for that specific community. And then Maria is going to walk you through the new portal. So the new framework that we have for the Common Metabolic Disease Knowledge Portal is the same framework, so software architecture and platform, we've applied to musculoskeletal disease. What's really nice is this is specific for a new community, new set of traits, but it's the same architecture, but we have it in its own resource so that people from that community can really use it to their benefit, as well as to the broader community. So real quick, those of you who haven't used this yet, the Common Metabolic Disease Knowledge Portal is open access and available to you all. You can get to it from cmdkp.org or hugeamp.org. We have two URLs that bring you there. And once you're there, you can peruse the various portals. One specific portal that you all know and love, I hope, is the Type 2 Diabetes Knowledge Portal. It is officially converted over. So if you type in the old URL, type2diabetesgenetics.org, it will redirect you here seamlessly. And you won't know the difference, except that it'll be much better and much more data and tools. If you want to go out to the Common Metabolic Disease Knowledge Portal at any point, you just click there and it will bring you to the broader resource that allows you to explore all the data sets and tools across the platform. So one thing that will be important to several of you who have used the Type 2 Diabetes Knowledge Portal for years and like some of the features that are still on it, there are two major features that are not in the Common Metabolic Disease Knowledge Portal yet that are soon to be in coming 2021. One is GATE. For those of you who know it, that's the custom analysis tool for aggregation tests. We'll be re-envisioning that completely for 2021, which will be very exciting. We'll include top med data, and we'll be expanding first. We're going to be top med data for the subsetted exomes of top med, but then soon thereafter, we'll be able to run custom aggregation tests on all of top med. And I think that'll be a remarkable coup for the platform. So that's two things. The other thing that's not in the new UI, that is in the old if you like it, remember it's on old data because we've updated to a lot of new data since then, is the variant finder. If you really like that, that's still there. If you're looking for the old portal, you just want to go to the bottom of the page on the new UI. And you'll see right here, you can get to a tutorial of the new UI, which is in the video right there, but also see the previous version of the portal will be right here. It'll be there for several months if you need to see it. However, if you have any citations that you've had in papers and things like that, we've all made it so those will redirect to the new site. So in other words, you won't have to have these bad URLs that don't work in your papers if you've cited the portal for any reason. What's nice is that this is here. We will decommission at some point, but it is there for those of you who still want to reference it. So couple things just about the integrated resource, just to remind you what we've integrated. This will be really helpful for when Doug and Maria talk about the musculoskeletal portal, because many of the same data types, actually all the same data types are represented, but for different traits. So I thought this would be useful to just walk you through real quick. So one thing about that, actually, before I dive in, the other three portals, the cardiovascular, cerebral vascular, and sleep disorder portal, again, are integrated in the Common Metabolic Disease Knowledge Portal. We have not yet redirected those. So those are still available on their old URLs, but we will probably do that in 2021. We just wanted to give them a little bit more time for that community to convert over. The portals are publicly accessible, but there's just different URLs at this point. And we'll do the same thing that we did for type 2 diabetes for these in the coming weeks, excuse me. All right, so integration, three main pillars of data: genetic data, functional genomic annotations, and results of bioinformatic methods. And these leverage each other clearly, right? So for your genetic associations, our rationale behind those, right, is to aggregate and summarize known associations for large authoritative data sets. And that's really what the Common Metabolic Disease Knowledge Portal and the type 2 diabetes before it was founded on, bringing definitive genome-wide association statistics and extensive phenotypes culled by the community. So the definitive way that they want to represent their phenotypes and credible sets. So that's really the foundational data set. And for that, as of today in the Common Metabolic Disease Knowledge Portal, we have data sets representing 50 countries. It's really quite impressive how many countries are represented in the combined resource. And we have 168 data sets across 325 traits for your perusal. In terms of functional genomic annotations, that's changed quite significantly as well, thanks to our partners at the Diabetes Epigenome Atlas. For these, the rationale behind these is to inform predictions of variant effects for cell types, tissues, and pathways of interest. So for these data types, these are housed at the Diabetes Epigenome Atlas, but they're represented on the portal in an integrated manner that you can query. And Maria will show you some of that as we go through the musculoskeletal portal. And for that, there are now a total of 3,529 genomic annotations across all of these tissues within DGA, spanning these large-scale annotation categories, and these are accessible in the portal to you as well. And so what's really nice about Kiel and his team is they've also expanded from just thinking about type 2 diabetes to all of common metabolic diseases, and also working with us to expand to the other disease areas like musculoskeletal disease. So they've integrated specific data sets that the musculoskeletal disease community has wanted. So this is really a testament to how this resource is growing. And because of its sort of nimble software platform, it's able to expand to common diseases quite readily, particularly if you have the expertise of the community helping you. And finally, results of bioinformatic methods, right? So this is the ability for us to integrate the genetic data and the genomic annotations and make inferences about the molecular cellular function. And there's a host of methods here. What we're really trying to do with the resource is integrate and implement methods that are sort of the most widely used methods within the field and run them at scale across all portal data. And I think that's a really novel approach. What's nice about that is we're not only doing that, but we're working with the methods developers, the partners, they're often collaborators, working with us to make sure we're implementing it correctly and reviewing the results of it, because oftentimes it's run on more traits than it's ever been run before. So it's kind of a neat opportunity for both the method developers and for us to represent these results to you on the portal. Again, some of these we're not the experts in, so that's why we call them the other people who are experts. And this is a work in progress. Much of the results that we're showing to you are either just getting in bioRxiv, recently published, not yet even published. So a lot of this is about bringing accessible results to you as early as possible and have them vetted by the community as well. So what's in the integrated resource, as I just mentioned, sort of pulling it all together across 77 million variants. You have the genetic data, the genomic annotations, and 4 large-scale methods we run across: VEP, bottom line association and analysis, genomic enrichments, and that's run with Gregor currently, and MAGMA, multi-marker analysis of genomic annotation. So those are what's in the integrated resource. And I think that's a nice stopping point for me to convert over to, so this same foundation is what you'll see in the musculoskeletal portal. You see the same sort of user interface, but specific for musculoskeletal disease. And what's nice about that is we've now partnered with a wonderful community of investigators in musculoskeletal diseases, the International Federation for Musculoskeletal Disease Research Societies. And they're working with us to bring together the definitive genetic associations, functional genomic annotations, and methods that are important to their community to represent those for their disease. So with that, I want to turn it over to Doug. He's going to talk about why musculoskeletal disease portal, and really from him as an expert, like his perspective on it. And then Maria's going to walk you through it, and then I'll do a little wrap up at the end.
Welcome to today's discussion, and I'm really happy to be able to tell you a little bit about where this portal came from. You will notice the unique icon of our musculoskeletal portal, complete with little bones, some soft tissue, and even joints. And that's what it's all about, because the importance of having a portal on musculoskeletal disease is perhaps somewhat unique from some of the other disorders. Just to highlight the importance here, musculoskeletal diseases are second most common reason that patients go to see their doctor. 18% of clinical visits led to a diagnosis related to bones, joints, and muscles, affecting 1 out of 2 persons in the US aged 18, and nearly 75% if you're over 65. And about 50% of the disabling conditions that plague humans are due to musculoskeletal disorders. It's the leading cause of morbidity in the workforce, and for those of you who are approaching retirement, it is the most common cause of early retirement. Some people have called it a global threat to healthy aging because it slows down, it removes your independence and your function, and a lot of suffering. Economically, big impact class of disease. Of the GDP, 5% went to musculoskeletal conditions, and a lot of workday lost due to this problem. Fortunately, the musculoskeletal diseases of interest for our portal are heritable. And here are some examples. This stands for bone mineral density, the biochemical phenotypes or biochemistries that indicate how the metabolic function of bone is operating is heritable. The geometry, non-invasive methods like ultrasound to measure bone, heritable. The fractures, heritable. You'll notice hip fractures ranging from 3 to 68. Early onset hip fractures are heritable, and as we get older, the heritability estimates drop. Hip osteoarthritis, heritable, and muscle, going from a 30-year-old to a 70-year-old, you can see the loss of muscle. That is not a random event, it is heritable. And potentially another factor that differentiates the musculoskeletal genetics area is that some of the diseases that we deal with, not to harp on diabetes, but certainly as a doctor, you almost need a specialty training to know how to decide which of the dozens of drugs to use for diabetes. I know there's a goal of preventing the occurrence of diabetes, but in osteoporosis, which is one of the diseases of interest, the treatment that is currently in practice is very suboptimal because there are fears of side effects that are quite rare, and doctors don't know enough about the disease. So as a result, their treatment's inadequate, the meds are not optimal. Osteoarthritis, there are no disease-modifying drugs at all. And some have referred to the muscle as the last druggable target in the body. But to date, there are no drugs that have been developed to prevent that age-related muscle loss. So because we all know that the types of portals that Noelle's talking about lead to ultimately to drug targets, this field is ripe for this portal development. Just to give you an idea, this is the most commonly prescribed drug on the market for osteoporosis. And these are the dates from 1996 when the drugs first came out until 2012. And there was an initial adaptation of, docs were prescribing, didn't get that, that high for very long because people started to stop taking their meds, and the doctors were not prescribing it because of fear of side effects, and the doctors don't understand the true impact of these diseases on their patients. So it's not a rosy picture for drug development in our field, and that's why the promise of these portals offers a lot. So how the portal came together was there is a federation of research societies that have come together in this field of musculoskeletal disease, teaming up with the Broad team to create the portal. And just to tell you what this federation is, the goal of the federation is to advance research globally to prevent and treat these musculoskeletal diseases, and collaborating internationally, sharing resources, raising public awareness, and educating. As a first attempt at bringing together the accumulating data from our musculoskeletal genetics work, both basic science and human genetics, a big so-called big data working group had been at least trying to make an inventory of data that might be useful for our research. And so we had this elementary inventory, but we, having seen the success and the beauty of the type 2 diabetes knowledge portal, we envisioned that a similar portal for musculoskeletal diseases could support experimentalists, not just be a listing of data sets, but actually a real functional portal like the ones you're accustomed to seeing from the group here. And one thing that the group decided was we were at another disadvantage compared to, say, diabetes, where people get human pancreatic islet cells and they do everything down to single cell transcriptomics. Well, the musculoskeletal field has been systematically excluded from all the big projects that you're familiar with, to comparably to other tissue types. So GTEx, there's no bone, there's one muscle, there's no cartilage. The Roadmap and ENCODE project, they might have one small bone cell, but it has been underdeveloped, making it difficult to use some of the standard epigenetic tools to map our findings. And we bring a very strong basic science expertise to the field, including mouse biologists as well as cell biologists. So we want to do what all the other portals have been striving to do by obtaining, curating, and integrating all of our unique data sets on this web server, a good interface that Noelle's shown you and Maria will show more of. And we're trying to promote the use of the portal. And we've gone out and started giving talks around the time it went live in March of this year. So I think that's pretty much the introduction. I hope you realize the importance of this fledgling portal that is growing rapidly thanks to the help of our colleagues here at the Broad. So Maria, I'm going to turn it over to you, and we can wait for questions.
Any questions for Doug?
Okay, I'll get started then. Thank you so much, Doug. That was a great overview. I didn't know a lot of that, so that was really, really useful. Okay, so I'm just going to give you a quick walkthrough of some of the highlights of the new MSK-KP. So this is the musculoskeletal portal in our new framework. And because we have this new framework that's so well developed and the data are stored in a way that they can be retrieved rapidly, it was pretty straightforward to implement the MSK-KP in this framework. And we're poised to do this for other disease areas too if we want. It's much easier now than it was before. So first of all, I just wanted to show you a little bit of what's in the MSK-KP. So here are the genetic association data sets that we have. And right now we've included phenotypes that fall within these 5 groups. And it's up to the MSK community to decide whether these are the most useful groups or whether they'd like to include some or omit some of these. And all the data are stored in one common database. And so we can customize any portal very easily by just choosing which phenotype group will be shown for genetic association data sets. So for musculoskeletal, we have a lot. So we have all these musculoskeletal genetic association data sets right now. And these are all the individual phenotypes that we have. So you can see we've got arthritis, osteoporosis, a lot of bone mineral density measures, whole body and also at various parts in the body. And we also have under anthropometric, we have a couple phenotypes that are relevant to musculoskeletal diseases and traits. And that's, for example, total body lean mass and appendicular, which is arms and legs, lean body mass. So we've got a good range of genetic association data sets that reflect musculoskeletal phenotypes. And of course, we're looking forward to getting more from the MSK community. We also have, as Noelle mentioned, epigenomic and computational data sets that we are getting through our partners at the Diabetes Epigenome Atlas. And there are too many to list on one page, but what we've done in this page is just to list some of the ones that the musculoskeletal community has requested specifically. So these data sets are in our database at the moment, and I'll show you some ways that you can access them. Right now, it's mostly accessible chromatin annotations such as ATAC-seq and DNA-seq, DNase hypersensitivity rather. Okay, so I will give you just a quick overview of some of the highlights. So when you type a gene name into the search box, you will go to a region page, and the region page is really the core of the MSK-KP, just as it is for the other portals in this new framework. And so this shows you the associations and the epigenomic annotations for this gene, the coding sequence, plus 50 kb in either direction. And you can expand the range if you want by just clicking on this button as many times as you want. And so the phenotypes, the associations across this region are just summarized in this part. These are the genome-wide significant associations, and then you can explore all of the less significant associations in this part. Now the page is set for a certain phenotype, and you can change that, but by default it's set to the phenotype that is most significantly associated in this region. So here it's estimated bone mineral density, or EBMD. And so that phenotype is what is shown, associations for that phenotype, in this table and this tool called the genomic region miner that I'll illustrate in a second. And so the table of top associations shows bottom line meta-analyzed associations. And this is actually almost everywhere in the portal. The associations you'll see are from our own meta-analysis of all of the data sets that we have to get one bottom line p-value for each variant and phenotype. And we're still working on this. For example, we know that we need to show the total sample size for each meta-analyzed p-value, and we don't do that at the moment, but we're working on improving this. And just to note that the table is filterable by lots of different measures, and if you filter the table, it also filters the locus zoom plot below, which is pretty cool. So if you want to only see missense mutations or something, you can adjust the locus zoom plot to only show those types. So let me show you how this tool works. This we're calling it the genomic region miner, mostly because it has a nice acronym, GEM. And this is a way to explore associations and epigenomic annotations across the region, and hopefully to prioritize variants of interest. Oh, and credible sets as well. So you can filter. I'm going to filter because there's a lot of data, and it loads more quickly if we add a filter first. I'm also going to zoom in a little bit, so you can't see what I'm doing here, but I'm holding my shift key and scrolling, and then you can really zoom in. You can also pan by clicking and dragging. Okay, and so I'm gonna just look at this variant. This is one of the most significantly associated variants. It's listed in the table above. And so let's add some annotations. Now, here's a caveat. Because there are so many annotations, this list is incredibly long. A lot of these are transcription factor binding sites, and they're listed individually by transcription factor. So we're actively working, and well, I should say our partners at Diabetes Epigenome Atlas, Kyle Galton and his team, are actively working on creating an organized, reasoned schema for these annotations. So it will be easy to find them. For example, we'll probably have a category here that's just transcription factor binding, and then you can choose the transcription factor. So we realize it's a little hard to wade through these at the moment, but one good thing about this interface is that you can just start typing and it'll filter it down. So we're looking for accessible chromatin, and let's choose this one. And yes, we realize Max and Max 2 are not super informative. We're going to work on explaining that. So we added accessible chromatin, and let's also add another chromatin accessibility, DNA hypersensitivity. So when you add these tracks, you get, okay, here it comes, you get tracks showing both of these types of annotations. And then you can explore these. When you mouse over them, it shows what the tissue is from. And these show the extent of the annotated regions. And there's a crosshair, so you can line up the annotations with your variant. And yes, so this is a bone relevant genetic association for this variant, and it happens to be in a region of open chromatin in, yes, typo, it should be femoral osteoblast. So that's of interest, obviously. This GEM module can help you find that kind of thing. If we look down here on the accessible chromatin, this was from the TAC-seq, we can see that it's also in an area of open chromatin in the humerus. So this is all consistent with what we know about this variant. But the point is that for variants you don't know so much about, this is a good way to explore their associations and the regulatory potential of the regions where they are. Okay, I don't have time to show you the whole MSK-KP, but I think I'll show you some highlights. So one of them is the phenotype page. So when you click on a phenotype name in the portal, there's often a selection to go to the phenotype page. So this is a page that summarizes the associations for this phenotype. And at the top, you see a Manhattan plot and QQ plot for all of our bottom line meta-analyzed associations for this particular phenotype. And then there's a table of the top associations for that phenotype. And also the gene level association. So these are computed using the MAGMA algorithm that Noel mentioned earlier. So it takes GWAS summary statistics and computes a gene level association score for each phenotype. And so this will tell you some of the top genes that might be relevant for this phenotype. And I'll show you in another tool in a minute. We have another module that will let you combine different phenotypes and find genes that are associated with multiple phenotypes. Okay, now this is all bottom line meta-analysis. You might wonder which data sets went into that. Well, on the phenotype page, you can see that. So here there are 2 data sets that were meta-analyzed to create these associations. And if you click on either of these, you'll go to what we call a data set inspector page. And that will show you the Manhattan plot and QQ plot just for this data set. And it will also have a description of the data set, a table of the top variants, and description, links to download summary statistics if that's available, and sometimes cohort information. So there's a lot of information about each data set there. And finally, for this phenotype, this table shows you the globally enriched annotations. And there's, again, we're going to be making this more obvious. But yes, so you can see there's some muscle, lots of muscle and skeletal associations for this phenotype, which makes sense. Okay, let's see. I think I want to show one thing on the variant page. So there's also a page for each variant and a page for each gene. I'm not going to go over all the details, but I did want to show one thing because we've heard from a lot of our users that they would like, they do want to know what the bottom line associations are for a variant. And the variant shows that across all phenotypes. But they also want to know what were the p-value and so forth for this variant in an individual data set. Sometimes people want to replicate their findings in one particular data set. So on the variant page, you can do that. So if you click on this datasets button, it shows all of the datasets that went into, in which this variant has associations. You can compare that. You can see exactly in which datasets and what the p-values were and so forth. So that's really handy, I think, for some investigators. Okay, I'm going to show you a new tool that we've just implemented, and this shows magma results. So we've run this magma method over more than 300 traits that we have, and so this is to compute using GWAS summary statistics to compute gene level associations. And so to use this tool, you can start by selecting any phenotype you want. You can also filter by p-value. But this will give you, this is like a Manhattan plot of these associations, gene level associations now across the genome, but you also get a table. And then the cool thing is you can add to this table. So let's see, let's look at, I don't know, hip osteoarthritis. I'm just pulling this out of thin air. So, but obviously people will have their own ideas of which phenotypes make sense to combine. And so you can just keep adding phenotypes to this and find genes that are associated with all of your phenotypes of interest. And that can be kind of informative, I think. Okay, I'm going to show one last tool here. So I think those of you who have been to our webinars before know that we've have been implementing methods for predicting effector genes from variants. And I think it was our last webinar where we went into these in some detail. But we've also done this in the Musculoskeletal Portal. Now, in our other portals, we have two other methods, but they only have predictions right now for type 2 diabetes. So that's not super relevant for the Musculoskeletal Portal. So we're not showing those here. But we are showing the effector index predictions that come from Brent Richards' group. They developed a method to predict genes using summary statistics and a variety of other DNase hypersensitivity and a variety of other data types. And they did the predictions for all these phenotypes. This is also going to be up to the bone community to decide if some of these may not be really relevant to show here, so they can tailor this. But some of them clearly are, like EBMD. So, and yeah, I'm not going to go into this in detail because I went over it last time, but basically what it is a method that results in a probability score that at a GWAS significant locus, this gene is the causal gene, the effector gene. And this gene right here, which has a high score, actually is known to be involved in bone development, so that's a good sign. And there's also a description of the method here. And so the exciting thing is that we are implementing this algorithm internally. And so we are going to be able to run this on all our data and for all our phenotypes. So it'll, even though there are not manually curated effector gene lists for every phenotype that we cover, we'll be able to at least offer some prediction. So that'll be really nice. Go ahead.
Maria was saying, I think that's exactly right, Maria. And I think the goal is, one thing that's nice about Brent Richards is that I think his research background is in bone and musculoskeletal disease. So, it's really nice to have that trait from him on the portal for musculoskeletal disease. But I think, given that these are predictions and some of them aren't published and some of them aren't vetted, what our hope with the platform is to do two things. One, to as Maria mentioned, implement it across all of the traits and represent the results, but also, work with the respective communities. For diabetes is a very large effort for us in the next 5 years to really, not only, improve the portal, but actually generate more data and tools with, thanks to the NIDDK for type 2 diabetes effector genes. But some communities don't have as much of an effort mounted. But our goal is to use the resource and to partner with the community to try and do any curated list they have, any inferences that they have. For example, cardiogram. We're going to work with cardiogram in the next couple of months. They have a gene prioritization method that they've implemented for cardiovascular disease. There's coronary artery disease specifically. We're going to be running that through the platform, but also representing the results for what they've done curated for those traits. So it's kind of a combination of running the method at scale, right? But also working with the community to tune that and represent the best results. And so our portal, kind of sort of spans the gap of being, a place where results are made accessible as fast as possible to the community, with the community backing it, but also constantly fine-tuned and represented to try and hone in researchers that are not in our field to make the best decisions about genes of interest. So really that's what this is as a scientific resource that we're hoping to really, grow in the next couple months. And when Maria's done, I have a couple slides, but I'll let Maria finish to kind of describe what we're doing next. Maria, are you done? You still got some more stuff to go through?
Yeah, I think since we're doing well on time, I'm just going to show the gene page really quickly because there are a couple things to say about that. We've made some differences. We've made some improvements since the last time. So, okay, I went from the home page, I entered a gene name, and now we're at the region page. But then if you want to see the gene page, you can click on the gene name. And also when you see a gene name other places in the portal, it'll take you to the gene page. And so this is just meant to be a place, where you can learn about the gene itself with links to a lot of external resources, such as Unicrot and others that will, tell you the function. That we are also planning to add more, informative, more information to this page that will give you an at-a-glance view of the gene product, such as gene ontology annotations. So we have a couple tables here. So one is common variant gene level associations for this gene. And again, these are MAGMA results. So there's a table of those. And then rare variant associations. And I want to explain this because several of our users have gotten confused about this, and it is a little bit of a confusing message. So in this new portal, we have a mechanism for loading data that are still private and allowing certain people to preview them before they are released in the portal. And so that's what you see here. This is, so these rare variant gene level associations come from the 52K exome sequencing study, or AMP-T2D genes exome sequence. And the first analysis, which is published, looked at type 2 diabetes associations. And so that's what you see here. However, there's an analysis in progress looking at 23 traits, or I guess 22 more traits, quantitative traits. And those results are loaded in the portal, but only the collaborators can see them at this point because they're still working on the paper. So as soon as they give us permission, those will be public and you will be able to see them. But some of you may be in this position where you've given us a data set that is not yet released, and then you may see this message and you will be able to log in and see your own individual results. And I see that Laura raised her hand. If you have a question about this, why don't we go ahead?
How does Laura unmute herself?
I see it. I got an unmute signal from that. Thanks very much. So I'm curious about when the portal presents gene-based results based on MAGMA results. I mean, that there are variants in the region, right? And it's a much less specific measure than having the exome variants, which still could lead you to the wrong gene.
Sure.
How, for people using this more naively, and not realizing that potentially doesn't mean anything about that gene, I haven't gone through the portal well enough to know, when any of these comes up, what caveats or what kind of cautions come to people about that to keep them?
That is an incredibly relevant question. We were just discussing that issue internally right now. Yeah, you're absolutely right. And a lot of people have mentioned this, that yeah, MAGMA definitely has its caveats. And we do some explaining here, but, but we don't yet have, list the explicit caveats. And you're absolutely right. We need to do that. Because if people aren't familiar with the method, they'll just say, oh, this gene is, this gene does this, it's the effector gene. And that's not true.
Right. I see people do that in papers also. Right. And so it's a wider spread. Somebody reading papers might not realize it either based on how it's used there. So anything you can do there would be fantastic.
Thank you. Great point.
Yeah, that's really the next sort of thing for us. As we've been able to make these more readily accessible faster, we have to catch up to the caveats and how to, I think, present the what and the why really clearly on the portal right away to the user, particularly if it's someone who's not, deeply embedded in the statistical genetics community. So that's what we're hoping to do in 2021. Mm-hmm. It's a great point. And actually, Laura, you'd be very useful in that. And, our sort of analytical team that we work with, you'd be really useful in helping us, get the right gist of how we represent that information to users. I saw some other hands up.
I'm real happy to be a part of that.
That'd be great. I saw some other hands up. Were there other questions?
Well, yeah, actually, that's about all I wanted to say about the gene page at this point. That we do have these gene level associations are not MAGMA. These are computed. Were computed in this study, and there's information here about how they were done. Yeah, I think that is really enough for now, and we can go into more things in the questions. But let's leave some time for everything else.
Questions for folks? All right, well, I'll continue on, but if you want to raise your hand, just go for it, and we'll unmute you and we can discuss it. So 2021, hopefully will be a better year for many other reasons, but it will also be a better year, I think, well, an exciting year for the portal. I just want to put some teasers in for you all in terms of what we're thinking about for 2021 in terms of large scale investments. So the first, I alluded to this earlier, which is we really, GATE, which is our genomic custom aggregation tool, which we worked very closely with our Michigan team on making this possible, is going to be totally refreshed and new. So the UI you've come to use in the old portal for type 2 diabetes will totally be re-envisioned and implemented in the portal in January of 2021, and will include major data sets that were already on the portal, the AMP T2D genes, 52K exomes, as well as top med. We're going to start with exomes because the genomes are super big, and we have to figure out very clever ways of working with that data and how to make that accessible. And actually, that's going to be an exciting foray for us because it will hopefully bring us closer to working with other groups and even other resources like Biodata Catalyst, because that's where the top med data is housed. So look for that exciting developments in 2021 for that. In terms of methods, big methods that are really relevant to the community, LD score regression will be implemented on the portal on all traits. I think this is a broadly used tool to add to our global enrichment tools in the portal. Also POPS, which is the polygenic priority score method by Hilary Finnegan and Al Weeks. That method is also been widely asked by our users and will be implemented on all portal data. What's nice about that is that's been sort of tested through and has some publications in place, not only for the method, but also for application to traits of interest, cardiovascular disease being one specific. So that will be coming in 2021 as well. We're also, one of the things for diabetes that we have not been able to do as well as we would really like to are represent results for complications in the context of type 2 diabetes. This is really important to NIDDK and really important to understanding the genetic architecture of type 2 diabetes. So we're going to implement a sort of a new tool to allow you to browse the results of a specific trait, not in the context of type 2 diabetes like MI, but then also in the context of diabetes. What's nice about that is we're finally at it's just starting to get to the point where I think we have enough data to make that a useful tool. So we're going to have that plus some data underneath that represented in 2021. Type 1 diabetes knowledge portal also bringing to, adding to the type 2 diabetes footprint in the common metabolic disease knowledge portal. We have that portal live and active on the common metabolic disease resource, but in 2021, we're really going to hit the gas on integration of new data types and representation there. You remember, while it is diabetes, it has an autoimmune component. So we're working with the collaborators and the scientists, that we're working with there, Steve Rich and Kyle Galton, to really figure out how to make sure that portal represents type 1 diabetes appropriately. Another exciting development, lung disease. I think we've talked about this before. We're working with Benjamin Raby and Michael Cho to work on getting a publicly accessible lung disease knowledge portal. And that will be in the new framework. And what's nice is we're going to be adding host genomic data for COVID-19 as well, integrated in that. I think is quite relevant to lung disease. It may be relevant to other portals. That's up in the air. If you guys want that, we will add it. But I think for lung disease, it's specifically a really great trait to add to a portal for lung disease, asthma, and the other traits associated. And also, so getting to some of the point that Laura raised about how we, walk people through our intuitions of the methods and the caveats. Really what we're going to add to the webinar series in 2021 is the science of the portal. A lot of what we've done last year is, telling you what we've done, how to use it. And I think that's useful, but really this is a scientific resource. It's a community. It's a lot of people who have contributed, and we want the science that the contributions represent to be represented to you as well. So if there's a specific data set or a method that we've really, integrated in the portal that we think is a broad use, we're going to bring that to you as our user base and share that with you from the scientists, from the method developers, so that you will see how to use it on the portal in the context of what it was meant to be. So that's really what we're looking forward to doing in 2021. Again, join us for our webinars. You already have. I'm loving that you're here. We also have our resources tab. We're starting to roll out new videos. Obviously, our old videos aren't as useful as they used to be. So we have a basic video on the new portal. We have documentation. All the webinars are publicly accessible to you. We're going to be doing tutorials and having our PDFs that describe the functions and different tools. So all that's there in the resources tab if you need it. And with that, I think we'll tend to draw today to a close a little early. If there's any questions, love to hear them. If not, we'll give everybody some 10 minutes back. Hey, Mark, do you have a question?
Yes, can you hear me?
Yeah.
Probably an overly simplistic question, but why not ask it? So I see for the individual variant associations, we have p-value, beta, odds ratio. Some applications you'd like to see standard error or 95% confidence interval. Mm-hmm. Is that something that's findable in this resource, or we'd have to go back and calculate it ourselves?
I think we used to have it. I'm trying to think if we don't have it anymore. Good point. Maria, do you want to draw up your screen? Or
I don't think we show it, but I think we have it. I think we could show it.
It might be in the old portal, actually, I think. Mm-hmm. So certainly in the forest plots, we have confidence intervals. Right?
Oh, yes, that's true. In the forest plot, we do. On the variant page, for example. Yeah.
So it is there. It depends on the particular view that you're looking at, Mark. So you have one particularly in mind you're asking about?
If you could just pull up any quantitative trait.
Sure. Yep. Maria, do you want to do that?
Oh, sure. Yeah, and I just heard from our engineer, Jeffrey, that we do have it. We don't show it currently.
Right. And what I love about the new resource is, and Jeffrey, because he's our lead engineer, these columns are much more straightforward to add. So if there's something you want to add in terms of a statistic being reported and we have it, we can add it to the table. So Maria, thank you.
Just so, for example, just like in this top single variant associations table, in something like this is where you'd like to see, yeah.
Yeah.
Or on the region page, we have a similar table. Yeah, so basically anywhere you would just like to see the standard error and confidence interval. Yeah. Okay. Makes sense. Great.
No, those are useful things to add. Yep. Okay.
Other questions?
Yeah, I thought you were,
sorry.
I thought you were going to show us how to get that.
Oh, we don't have it currently. No, we'd have to add it. Yeah.
I see.
Yes. So I think it's in the old, I think we have it represented in the old portal. I don't think it's on the new one. There may be some reasons behind it that I don't fully understand in terms of what we chose to represent there, but we can find that out for you, Mark.
Well, so
yeah, go ahead.
The forest plot does graphically show confidence interval.
Yeah.
Okay. Great. Well, I'll be in touch if I have questions. Thank you.
Thanks, Mark. Any other questions?
Yeah, this is Phil Smith. I had a question. So I'm really excited about the type 1 portal, and you mentioned autoimmunity and, let's say, inflammatory biology writ large. And obviously this is an area where there's a tremendous amount of genetic data. And I'm wondering if there's any discussion about bringing that particular community into the portal.
That's a great point. That's what we've, so Kyle and Steve and I have begun to discuss that, because, you know, I think at the Broad, we have some strong communication, some strong roots there with IBD, with Romnick and Mark Daly. So that component is definitely a piece of what we've been thinking about. And obviously Jesse Engritz, who is, you know, a member of our AMP community, he works with IBD as well. And I think, so from that perspective, we're trying to figure out how we might integrate that. Steve and Kyle, you know, given their sort of sense of the type 1 diabetes space more, also, you know, have contacts in that space. I think what's interesting is to figure out how best to do that, right? You have an AMP in, you have many different possibilities, right? You have an AMP in rheumatoid arthritis and lupus, which are also autoimmune diseases. So that might be an avenue by which we partner. But I think for us, just on the ground, I think we're considering some very closely related autoimmune disease to start with that we might bring into the portal and represent and have as an option, and just start with simply representing it and see if we can get, you know, people from the community to buy in. And if you have any ideas, that would be useful.
I mean, so one obvious idea is celiac disease. And, you know, we actually have data in the Teddy study that relates to that. And
that's a great idea.
So it's sort of a entree into the IBD folks, maybe.
That would be nice. Yes, I like that idea. Okay, cool. I'll talk to Steve about that because he might, because he's obviously, you know, he heads up Teddy. That's great. Yeah. All right. Jason, I saw a question from you about an API. There will be, not yet. We have to update our APIs. It's a great question. And I don't know about our horizon for that. I'm hoping early in the new year. I think that's actually probably an accurate statement, but I will get back to you on that, Jason, in terms of where that is. The current APIs are still available, but they're a little outdated with data. Any other questions? All right, super. So our next webinar is going to be in January at noon, and we'll send around an agenda for that and let you all know about that. And thank you all for joining us. And in terms of just programming announcements, the Musculoskeletal Portal will be, you know, transitioned over to the new UI very soon in 2021, but you can still find it. If you type in the URL, you'll get to it. It's publicly accessible. And, you know, this will be available if you missed it or you want to share it with anybody. And if you need slides, just give me a holler and we'll share them with you. Thank you, Doug. Doug, you've been awesome to work with in the last couple of years. You really, it's really been great to learn about the community through your eyes and to be able to have this opportunity to work with the musculoskeletal community more closely. So it's been really wonderful.
Same from our side too. And just put out a faintly disguised ad for any data sets where we're still combing the area for data sets. So we're trying to create some sort of system so we can evaluate what's out there. We think we have a good handle, but if you're on the call today and want to contribute, just let us know.
Excellent. Phil, what did you want to say?
Yeah, Doug just raised an idea in my head, which is how are we outreaching to all these communities that we're now embracing? Is Doug planning to give a talk at the you know, relevant meetings, or are you planning to go on the road for the various research communities we're now embracing?
A little bit of both. So I think when there's a scientist in the community that has like the right pulse, like Doug, I think, you know, I like to let him lead. He understands the community and sort of, we come along sort of, you know, be the demo force. I think so we're really actively reaching out to the cardiovascular and the cerebrovascular communities because those are intimately close to them. Type 1, I think, you know, Steve's been really great to help me kind of navigate that with the right collaborators. I think that's one place I'd like to get in front of more folks. In terms of like what data sets would be relevant to bring in and just make them more aware of it. I think that's one place where we have some work to do. Obviously the portal's just been made live, but still I'd like to do that. We're really focusing, 2 of the communities really want image data, specifically for sleep disorder and cerebrovascular. So we're actually writing a grant with Jonathan Roseanne and some folks, Dan Marcus, who invest in, you know, bringing CT and MRI and imagery scans for cerebrovascular disease and making those accessible as well. So it's sort of another way of deeply phenotyping, you know, cerebrovascular disease and other events that require imaging. So
yeah, heart failure is on that list too. So heart failures get a lot of imaging data. That's
even better, heart failure, and that's much more relevant to our particular experience. That's great. I like that. That's a good point. So that will be useful for that.
Yes.
So I think it's a combination of going to meetings, partnering with the scientists. One of the things that we've been toying with, and I think Jason and I would like to do this as well as 21, is create an advisory group around this resource, not just for diabetes, but really think about like the people in the communities, having representatives from just the disease communities, the methods community, and you know, from visualization world. And have those people at least meet regularly. You know, obviously, it's, you know, coalition of the willing there, and to advise us on the best ways to do certain things. Each disease obviously is going to have their own, you know, needs, and we want to maintain that. But it's more about giving us some people to bounce things off to more routinely. I think that's what we've created, a critical mass, in my opinion, such that will be useful to the community, particularly if they want to. We want them to take us seriously and vice versa, right?
Yep.
So I think that's our goal. So anyone has ideas or would like to, I'd love to hear them, because, you know, this is sort of exciting new territory for us.
It is great. Really appreciate you doing this.
Oh, thanks, Phil. Thank you, everyone. Thank you, Doug. And we'll see you all in 2021, where I hope the world is a more smooth place for everybody. So I wish you all the best. Take care, everyone.
Thanks, everyone.
Bye-bye.
Bye-bye.
Bye.